I posted your test scores because I wanted you to have them for drop day. So I'm preaching to the choir because you're still here, right? You guys did almost exactly the same average as you did on the first test, which was 5 points higher than the other class. We had both really, really high scores, hundreds, good job, and several A's, and we had really, really low scores. I can't remember if this class had a 20-something in it or a 30-something in it. Okay, so if, however, you're one of those people who's just not doing as well as you want, and you haven't come to see me, you should, because I will take the time, after you've taken the time to look through your test, I will take the time to help troubleshoot your study techniques so that you can increase your likelihood to do well. I won't give you answers, right? But I don't know what's going to work with an individual, but I have seen a lot of students be successful and not be successful, and so I can share some of that with you. And I'd be happy to do it if you're going to take the time. Now, I'm not going to give your test back today because I've still got 2 tests of DRC that I need to go pick up. Give them back on Tuesday, but you can still come to my office hours after this, and then you can see it there. I just don't want to like have 2 people not have their test because that's kind of mean. And I didn't tell you I'd have them back until Tuesday anyway. Okay. So, did you get my announcement that we're going to do synapses? Okay, good. So we're going to do synapses before we do the other stuff. Synapses are my favorite, so I'm very, very excited about this. So we're going to do synaptic transmission and neurotransmitters. Most of it's pretty damn logical, and then there's also a lot of stuff that we'll talk about related to drugs, disease, crazy, while you're smart, while you're happy, while you're everything, which is specifically why I like this set of notes. So first thing is, usually, most semesters I teach this right after action potentials because that's where it goes, people. But I was trying to hook it up so that you guys could have endocrine at the same time you had endocrine in lab. So we're going to go back, and basically what we're going to do is we're going to fill in the rest of the story from after an action potential. So I'm going to show you this animation a couple of times today. So basically, you know that we have an action potential, and then what we are going to do is talk about the mechanism of releasing a neurotransmitter, which is what you're about to see right here, okay? And then we are going to talk about how the neurotransmitter causes activity on the postsynaptic cell, yes? And then we are going to do a tour of the major neurotransmitters. We're not going to learn all of them because we don't even know what all of them do yet, and there's more than 60, but we're going to learn the ones that I think you're going to run into, either being a human with a brain or in your career. Okay, good? So we're going to come back to this a couple times. So there isn't much before you begin on this set of notes. It's basically, do you remember what exocytosis is, what a gap junction is, what resting, graded, and action potentials are? Hopefully you do. And depolarization, hyperpolarization, all that kind of stuff. We're going to go ahead and go to what is a synapse. And I hope that in anatomy you talked basically about what synapses are. Synapses are really the site of communication between a neuron and the cell it's talking to. It's called a true synapse if it's neuron-to-neuron communication, yes? But we also know what it's called if it's neuron-to-muscle communication. What's that called? Neuromuscular junction. Yeah, we already learned about those. We know how those work pretty well. And then it's called a neuroglandular junction if it's between a neuron and a gland. This set of notes really concentrates on true synapses, neuron-to-neuron communication. So we will talk briefly about the other kind of stuff, but okay. So what a synapse is the junction between a neuron and a neuron or a neuron and an effector. And so a couple of interesting things about synapses. They are definitely the site of communication between neurons. And one of the things that people have figured out over the last few years studying neuroscience and neuroplasticity is that we know that you can actually form new synapses based on what you do with your brain. You don't seem to form any appreciable amount of new neurons after about year 2, when your brain is pretty much baked. But you can form new synapses. So what could happen in this situation is, let's say that you get some kind of exposure. Let's say it's in your cerebral cortex and we're trying to form some kind of intellectual connection, right? Between something, for instance, that you learned in anatomy and something you learned in chemistry and then stuff we're doing here, which happens a lot in phys, right? Phys is like the Venn diagram of where those courses like 1610, anatomy, and chemistry all come together. So what can happen is you can form a new synapse. This guy can butt off an axon collateral and actually form a new synapse to here, right? And what that could do is either go to a neuron that it wasn't attached to before and form a connection, or form another connection with the neuron that it was already attached to. If it forms 3, 5 connections to the second neuron, then are you going to get communication between those two faster or slower? Faster, right? Because you're going to release neurotransmitters and cause a faster response. So interestingly to me, we have referred to learning things as making connections for ages, but that's actually what's going on in your brain. And we had no idea. We just happened onto a metaphor that actually what seems to happen in your brain and what's going on a hell of a lot this semester is that you are actually forming new synapses, making connections in your brain. So a new synapse is analogous to a memory. Now, it's not as simple as one synapse equals a memory, right? But when you are forming memories, you are actually doing synaptic plasticity and forming new synapses. And that goes on all through your life. It goes on really massively when you're tiny, yeah? And then as you get a little older, it actually starts pruning your synapses so that you start refining the ones you're going to use over and over again. And then as you learn more things, you actually form more synapses. Most of that happens when you're sleeping. And somebody in the last class asked about this, and I thought it was a really interesting question, so we'll go ahead and address it. Do you ever cut back your synapses? Do you ever prune your synapses? Yeah, a lot of what adolescence is pruning synapses. So when you are a kid, you're forming tons and tons of synapses, all of the places that your brain could go, right? Forming all of these synapses. But with adolescence, you actually decrease the number of synapses and reinforce the ones that you use over and over again. So for instance, in adolescence, you very often define the kind of person that you assume that you are going to be. Like if you are a punk rocker, you're going to reinforce punk rock, right? What are you going to do to your classical music synapses? You're going to let them wither on the vine. That's what you're going to do, right? So you are like really reinforcing the ones that you use over and over again and pruning the ones that you don't. Which is why, like for those of us who are parents or are going to become parents, or some of us might even still be in adolescence, expose your kids to every single thing that they could possibly be interested in and have a passion for as they are going through adolescence, because that is the best time for them to reinforce those behaviors that are going to end up being permanent. So if, for instance, you wanted them to pick up an instrument and be a musician, it needs to happen not when they're 40. When is the most creative time in your life? When is almost all of the best art made? Teens and 20s. Yeah, when is almost all of the best music made? Teens and 20s, right? Now, everybody thinks they're jerks, but they're just really creative people who are trying to figure out who the hell they're going to be. So that is sort of synaptic plasticity, that's what they call it, your capacity to form new synapses. So as far as the types of synapses go ahead.
Is that why when there's an adult has an injury and have to relearn to do basic things like walking or speaking, is it because those synapses have been improved when they were younger?
Yeah, it's much harder. First of all, your synaptic plasticity decreases as you age. It's really, really major when you're little, especially when you're an infant. And then as you get older, it's harder to form new synapses. And a lot of the pathways that you would have used to do those things have been trimmed down. So yes, you've got a couple of things. You're less likely to form new ones, and then you already reinforced specific pathways. So it's going to take a little more for you to do it. Okay, so there's two basic categories of synapses. Electrical synapses are when two cells don't release neurotransmitters. They're just stuck together with gap junctions. And it's really fast, but it's a one-trick pony, right? Because whatever cell A does, cell B has to do exactly the same thing if they're stuck together with gap junctions. And think about this, guys. Do you want all of your neurons on at the same time? Hell no. That is batshit crazy. Like half your day is spent trying to shut up some neurons, right? You're like, no, shut up. I don't want to think about that thing that happened in 7th grade. And also you shouldn't punch this person or grab that boob, right? Half of your day is spent doing that, right? And thank goodness it is. We appreciate that. Thank you. Okay, so electrical synapses are gap junctions. They're not very common in humans with the exception of like cardiac muscle and smooth muscle. FYI, you do have a whole bunch of them in your nervous system embryonically because like your nervous system is put together as like a scaffold and it's put together with gap junctions, but then as it matures, you pull them apart and start using synapses, like regular synapses, chemical synapses. So the type of synapse that we're going to be talking about in this set of notes is a typical synapse called a chemical synapse, and the chemical synapse communicates between cell A and cell B by releasing chemicals, yes? Into the synapse, yes? Makes sense? So, and of course, the type of synapse we're going to be talking about is a true synapse, neuron to neuron, and there's also neuromuscular junctions and neuroglandular junctions. So we are going to move on to true synapses. Okay, so what we have, and you guys have enough anatomy and physiology knowledge already to look at any two neurons in series and tell me which one is the presynaptic neuron and which one's the postsynaptic neuron. You can do that, right? So which one's presynaptic in this picture? Top one. Okay, so I'm going to draw it sideways, okay? And we are going to identify some players in this process. Okay, so which of these is the presynaptic? Yeah, this one's pre. A good habit to get into when you're talking about anything related to synapses and neurotransmitters is don't say neuron without identifying whether you're talking about presynaptic or postsynaptic, because we're going to see that different things are going to be happening on each one. Okay, so presynaptic and postsynaptic neurons. So we're going to start learning what happens with these. So let's name a couple more of the players. So this is the presynaptic neuron, the postsynaptic neuron. We can identify the bulbous ends right here. These are called axon terminals, or sometimes boutons, or whatever the hell you want to call them. And they have vesicles stored in them. These are synaptic vesicles. What's in there? What's inside the synaptic vesicles?
Neurotransmitter.
Any neurotransmitter. It could be acetylcholine, but yes, any neurotransmitter. So, and what is going to happen is we will see that it is going to be released. The neurotransmitter is going to be released. This is your neurotransmitter. Okay, so what else are we going to have? So we've got the synaptic vesicles, the presynaptic neuron, this space between the two. This space in here is filled with extracellular fluid. It's called the synaptic cleft. And I want you to distinguish that from the synapse. Okay, so the synapse includes the whole region where the pre and post are communicating, but the synaptic cleft is just this little space that you diffuse across. Does that make sense? So which one is a broader term, synapse or synaptic cleft? Synapse, yeah, synapse. Okay, so now I want to talk about a couple of things. So I want to use a metaphor here, okay? We are going to use pitcher, catcher, ball metaphor, or baseball. Ready? Okay, so synaptic transmission is going to be one way. What we are going to do is we are going to release a neurotransmitter from the presynaptic cell. It's going to be caught by a mitt on the postsynaptic cell. What do you think the mitt is?
Receptor.
Yeah, it's going to be a receptor. Yes? Does that make sense? Okay. So the presynaptic cell is the pitcher. It is going to throw the ball. What's the ball? Neurotransmitter. That is going to be received by the mitt. Yeah? And the mitt is the receptor. And the postsynaptic cell is the catcher. Right? Got it? Okay. So here's the important thing. The important thing for you to understand is that synaptic transmission is one way. It never goes back, and here's why. Even if the catcher gives the ball back to the pitcher, it can't cause a response because the pitcher doesn't have a receptor. Do you see that? So even if, and sometimes it does, the catcher gives the ball back so that the pitcher can use it again, it's not going to send an action potential that direction because that side of the synapse does not have a receptor that can cause a response. Does that make sense? So synaptic transmission is one way because the presynaptic cell does not have a receptor at that site. Does that make sense to you guys? Okay? So that means that I can never get an action potential going that way on that side, right? It goes that way, causes the release of neurotransmitter. Even if I roll the ball back, it's not going to cause a response. Does that make sense? So it's one-way transmission across a chemical synapse. Everybody good with that? Okay, so transmission across a chemical synapse is always one way, exclusively one way. I don't know of any examples or any exceptions on that one. Okay, so the function of a chemical synapse, now let's look at it in words and make sure that we get into some details here. So a neuron is separated from its neighbor by a synaptic cleft, yes? And the action potential, and this is important for you to think through, the action potential cannot jump across the cleft. So if I had an action potential here, yes, it's going to go that direction. Let's say I want to start an action potential here. It can't jump across the cleft because isn't an action potential an event on a cell membrane? Yes? It's a depolarization on a cell membrane. Can you have an action potential in the absence of a cell membrane? You can't. So you can't jump across the cleft. What you have to do is start the damn thing over every time. Every single time. How did we start it on this side? We had some kind of chemical binding to chemical messengers trying to depolarize the threshold, and then we started an action potential. How are we going to start on this side? Some kind of chemical binding to receptors trying to depolarize the threshold so that we can get an action potential. So you can't jump across. You have to restart it. Does it make sense to you that for that reason, this is the slowest part of the nervous system? The synapse is the slowest part of the nervous system. Good. Okay, so the space is bridged with chemical messengers, and you guys know, but say it anyway. What type of chemical messengers are we dealing with? Neurotransmitters, exactly. When the action potential reaches the axon terminal down here of the presynaptic cell, you are going to open a kind of channel that we met briefly last time. And these are voltage-gated calcium channels. So listen, those open in response to depolarization, and they are only at the axon terminal. I actually drew them too far up here, but I ran out of room. They're actually down there at the bottom. Okay, so listen. They open in response to depolarization. So are they going to open at the beginning of the action potential or at the end? At the end. They might open in response to 0 millivolts. Yeah? And what's the last part of the cell to get depolarized? Yes? All the way down there. That's the last part of the cell to get depolarized. So what happens is when those open, the voltage-gated calcium channels open, calcium is going to move down its electrochemical gradient into which cell? Into the presynaptic cell. The increase in calcium concentration is going to be the trigger for exocytosis. Does that make sense? So I'm going to show you an animation of that. So let's look at the same animation that you guys already saw. Okay, so watch. What is going to happen is the action potential, watch, action potential is going to come to the axon terminals, open voltage-gated calcium channels. Calcium is going to move down its electrochemical gradient into the presynaptic cell. That increase in calcium, as long as you have ATP for exocytosis, will cause exocytosis. Okay? And then the neurotransmitter, whatever you had stored there is what is going to be released into the synapse, right? Generally speaking, a neuron makes, stores, and releases a single neurotransmitter. We did find one like 4 years ago that makes, stores, and releases 2, and literally nobody understands how the hell it would know which one to release. So we're going to pretend that one doesn't exist. So most of them, like this would be an acetylcholine neuron, always releases acetylcholine. Okay. So when the action potential reaches the axon terminal of the presynaptic neuron, then the voltage-gated calcium channels open on the presynaptic neuron. Calcium moves in from what fluid to what fluid? ECF to ICF of the presynaptic cell. Now, every single neuron that I know of uses voltage-gated calcium channels to cause exocytosis of neurotransmitters. Remember me telling you before that calcium composition is really, really important for neurotransmission, right? If you have a calcium problem, you've got neurotransmission in damn near every axon terminal in your entire body. So I don't know of an exception. There probably is one. So it's really important everywhere. So calcium moves into the presynaptic neuron and causes excitatosis of whatever neurotransmitter was there. Some that we will learn, acetylcholine is this one. GABA is another one. Gamma-aminobutyric acid, we'll learn about that one. Epinephrine, norepinephrine, dopamine. We're going to learn not 60 neurotransmitters, but we'll learn the ones that you're going to run into or take drugs for over the course of your life. And whatever neurotransmitter is there is going to diffuse across the cleft, and then it is going to bind to receptors on the postsynaptic cell. Those receptors are usually, but not always, ligand-gated ion channels. Yes? Okay. So if you bind to and open a ligand-gated sodium channel, then you are going to blank polarize the postsynaptic cell. Depolarize the postsynaptic cell, exactly. So the neurotransmitter is going to bind to protein receptors on the cell membrane or dendrites of the postsynaptic cell. By the way, this one is one I won't ask you about either, but FYI, usually you form a synapse at the dendrite or the cell body of the postsynaptic cell, which makes sense because that's where you have most of the receptors, right? But some knuckleheads do this. I don't know how the hell that works because most neurons don't have receptors on the axon, but apparently some of them do. So we're going to ignore that too. Okay, so now, the receptors that we are looking at in this animation are called ionotropic receptors because the receptor is also an ion channel, so they call that an ionotropic receptor. They're really fast. They change the electrical nature of the postsynaptic cell really, really quickly. So that's what we're looking at right here. And there are two different categories of ionotropic receptors and of synapses that we're going to run into. So you do not always want to turn your neurons on. Sometimes you want to actively get them closer to an action potential, and sometimes you want to turn them off or down. Does that make sense to you? Like right now, in your brain, you are turning on some neurons and you are turning off some neurons. So how do you do that? There's two different categories of synapses, and it primarily depends on the receptor that you have. An excitatory synapse is what you are seeing right here. So when the neurotransmitter binds to and opens a sodium channel, for instance, it is going to depolarize the postsynaptic cell and get it closer to threshold, yes? So that would be considered an excitatory synapse. Does that make sense to you guys? Excitatory. Now, it doesn't mean you always get an action potential, but it's going the right direction. Does that make sense? Mike, you have a question. What's the question? Not ready yet?
No, I'm not ready.
Okay. All right. So if the neurotransmitter binds to and opens a sodium channel, it causes depolarization of the postsynaptic cell and excitation of the postsynaptic cell. Is this question for you? Is what is happening right here on the postsynaptic cell, is this a graded potential or an action potential? It's a graded potential. It's a depolarizing graded potential. It's only an action potential if you get the axon hillock depolarized and open your voltage-gated channels. So all I'm saying is on the postsynaptic cell, I'm causing a depolarizing graded potential, which is excitatory, right? It's the right direction. Go ahead.
Okay, here we go. So action potential opens up calcium.
Action potential opens up voltage-gated calcium channels.
Right.
Yes.
And then exocytosis.
Exocytosis, yes.
And then that releases like calcium?
No calcium.
Okay.
Releases the neurotransmitter.
Neurotransmitter.
Yes.
But so in the picture
Picture or animation?
or animations.
Okay.
So is the neurotransmitters the one?
It's these little guys right here.
Yeah, that.
That's acetylcholine.
To the sodium gated.
Yeah.
Yeah.
The chemical gated sodium channels are ligand gated. Yeah. The neurotransmitter binds to the ligand gated sodium channel. Got it? Okay. So what is happening here? And if this were the same kind of synapse, what would be happening here? You describe what's happening on the postsynaptic cell as an IPSP. Break it down. It's not that hard. Excitatory, because that's what it's doing. Postsynaptic, because that's where it's occurring. Potential. If I could put an extra letter in there, I would call it an EPSGP because it's a graded potential, an excitatory postsynaptic graded potential, which is the precursor to an action potential potentially, yes? It's not going to get there every time, but it's definitely going the right direction, right? If the EPSP hits threshold for this cell, then I'll get an action potential. Is that okay? Got it? Questions? Okay, so instead of me telling you, I'm going to actually see if you guys can do it. How could I potentially, let's say this is a different synapse because you can't use the same synapse to do two different things. How could I actively inhibit this cell? So I'm going to release a different neurotransmitter, and then I'm going to tell you that the important thing is this right here.
What type use a blocker?
What kind? Well, you could use blocker, but I'm talking what type of channels. What type of channels would be inhibitory? Yeah, exactly. Good. Does that make sense? So if at a different synapse, yes, I released a neurotransmitter that instead of binding to and opening sodium channels, bound to and opened potassium channels, right? Then what would I be doing to the membrane potential? I would be moving it not closer to the threshold, but further away. I would be actively inhibiting what that cell does, okay? So that is an inhibitory synapse, and what an inhibitory synapse would look like if you measured it is this. So neurotransmitter binds to and opens not sodium channels, but potassium channels. Potassium moves out, you hyperpolarize the cell, right? And what is happening on that cell is an IPSP. Inhibitory, yes. Postsynaptic potential, yes. Graded or action? Graded, yes. So this is how you can actively turn off your cells. FYI, for those of you who care about such things, you can excite with a different ion than sodium. Don't listen to this if you barely got it. You could use calcium to excite because it would also move in, right? You can inhibit with a different ion than potassium. You can use chloride because chloride will move out, right? So you could do, right? But generally speaking, to get everybody started thinking about things that we're really familiar with, a very, very common way to excite is binding to and opening the sodium channel and depolarizing. A really, really common way to inhibit is binding to and opening a potassium channel and hyperpolarizing. Okay, you first and then you. Go ahead.
Why would you?
Because you need to right now. So your neurons, your entire cerebral cortex, at some point you are going to have to turn down some things. So right now, let's think about it right now. Right now, sex drive, raging in the middle of your brain. Not the time, is it? Not the time. Shut the hell up, I have things to do, right? So, right? So you spend an awful lot of time, or aggression, right? If I piss you off, you're like, I would really like to smack her. It's not the opportune time to do it. Please don't ever do it. I need you to inhibit a lot of your behaviors. A lot of social appropriateness is inhibiting your behaviors. A lot of it. So you have to turn some things off all the time. Go ahead.
I have two now.
Okay.
So first, you're saying that you can use anything to depolarize or hide.
Not anything, but a positive ion that would move in,
Positive or negative ion.
or a negative ion that would move out.
Oh, okay.
Yes.
So, and then for this inhibitory synapse,
Mm-hmm.
you're talking about something that's like conscious. Are these like always going on?
Yes, they
the
are.
autonomic is like,
Yes.
oh, you need this little thing to change this.
Absolutely.
So there are like thousands of these happening at once.
All over the
That's so much energy.
Yeah. Yeah. You are constantly turning some things down and some things up. Yes. I was only talking about the consciousness portion because you can you can understand that based on your experience, but most of your inhibition is actually going on subconsciously, far lower than consciousness. A lot of that is going on. Does that make sense to you guys? Okay, so an IPSP, and so let's reinforce this by saying an IPSP is an inhibitory postsynaptic potential. The most common type of IPSP would be started with a potassium channel. Okay, does that make sense to you guys? Okay, so let's reinforce that by talking about epilepsy. Okay, so I've learned a lot about epilepsy over the last year because I have a student who's been really open with me and taught me a lot about her epilepsy and her treatment. One of the things that we can use to treat epilepsy, and epilepsy can occur because of genetics, it can occur because of injury, infection, any number of things. It's a condition in which your neurons fire action potentials abnormally and inappropriately, and it can end up going across both parts of your brain. And of course, it can cause muscle contraction, right? Because your cerebral cortex controls your muscle contraction. So one of the first tiers of treatments that they use to treat epilepsy and prevent seizures is a drug called primidone, and this drug hyperpolarizes your neurons. Yes? And so instead of your neurons sitting around at maybe minus 70, they would be sitting around at minus 80. So you would be less likely to have inappropriate action potentials, but you're also less likely to have appropriate action potentials. So adherence to this drug is really pretty low because you feel like you can't think, you feel like you can't get out of bed, you're not having seizures, but you're also not doing a damn thing. And you feel like you've lost your mental acuity, your intelligence is diminished, your zest for damn near everything is diminished. And so people go on the drug, the seizures diminish, and they're like, okay, I'm not having seizures anymore, and then of course they go right back off the drug. And you can empathize, right, with that. Because if you felt like you were exhausted all the damn time and lost 15 IQ points, you'd be like, well, maybe a seizure every once in a while is okay. I don't think that kind of thought process goes into it, but it's unpleasant to stay on these drugs. So do you understand IPSPs and EPSPs? Got it? Yes? No? Go? Yes? Go ahead.
When you were talking about like what's appropriate behavior-wise,
Uh-huh.
would that have anything to do with like mental disorders?
Oh, yes. An awful lot of it. It's your prefrontal cortex that is primarily in inhibiting your behaviors all the time. Like a really large part. If you just think about your prefrontal cortex, it really doesn't complete development until about 24 to 27, right? Okay. And I want you to think about a toddler, right? Versus an adult. Somebody takes a toddler's fire truck. Their prefrontal cortex is not baked at all. What are they going to do? You're going to hit you, and they might even bite you, and then you get sent home, right? Okay. Now you're an adult, right? You're 28. Somebody takes your shit. What do? No, you don't. Mike beats him up. You think very carefully about ways to get your crap back that won't end up impacting your life negatively. So you are inhibiting certain behaviors because there are better, long-term goals that are more important to you. Go ahead.
Does it ever backfire if you inhibit so much? Like,
Oh, yeah. We will talk about a lot of drugs that people like that are really, really inhibitory, that chill you the hell out to the point that you are over-chilled, right? Like benzodiazepines, like Klonopin, those kinds of drugs. So, yeah, there's appropriate levels of chill and there's inappropriate levels of chill. And we can talk about those as we get into drugs and neurotransmitters. Okay, so now there is another type of receptor that is slower. Don't worry about it excessively, but I do want to tell you that ionotropic receptors like these are really, really fast to cause whatever response you want. There is another category of receptors that is really comparatively slow. I have no blasted idea how these evolved. These are called metabotropic receptors. I want to show you a little bit about them. We will see them again. They actually are not uncommon, but they confuse the living hell out of me because I don't know how they evolved. Okay, here's the deal. This is the ion channel that you want to open or close. But instead of binding to and opening or closing the damn ion channel, which would be a direct shot into the pocket, right? You decide you're going to show off and do the most complex combo shot ever. Here's what has to happen. You bind over here. These three proteins slide through the cell membrane, slap into that enzyme, makes a second messenger that then opens that ion channel. In Texas, we call this going around your ass to get to your elbow. Like WTF, right? I don't even understand how that occurred, right? It seems like evolving 1, 2, 3, 4, 5, 6 proteins to accomplish what one could do is confusing as hell to me. And somebody was like in the last class, like, can you tell me what the value of that is? I'm like, no, I can't. I don't understand how it occurred, but they're not uncommon. Can you see that these would be much slower to cause the postsynaptic cell response? Yeah, can you see? These are called metabotropic receptors. And they are important receptors for some things that you're going to run into. And you do not have to understand the mechanics of how they work. Right now, understand that they are much slower to cause the same postsynaptic cell response and involve way more proteins in the postsynaptic cell membrane. So those are called metabotropic receptors. Okay, so but we're going to, for simplicity's purposes, assume that most of the time we're talking about ionotropic receptors, which are easier for us to understand. Okay, so what is going to happen on the postsynaptic cell, guys, is the neurotransmitter will start some kind of graded potential on the postsynaptic cell, right? Could be excitatory or inhibitory graded potential. And if it was excitatory enough, it could start an action potential on the postsynaptic cell. Yes? That makes sense? Okay, so let me talk to you about one other thing that has to happen. And the other thing that has to happen is, so I want you to think about this. Remember frequency coding? So how we code in the nervous system for intensity is by frequency. Does it make sense to you that the postsynaptic cell couldn't understand or hear frequency coding if every time I released the neurotransmitter I left it there? So what you're going to have to do is remove it after it's used every time so that you can hear it the next time. Remove it, hear it the next time. Because if I just released it and left it there so that it could continue causing the postsynaptic cell response, I wouldn't be able to tell the difference between zap and zap, 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 zap. Right? Does that make sense? So, just as important as causing the postsynaptic cell response is removing the neurotransmitter after you've caused the postsynaptic cell response. Does that make sense? Okay, so what are the ways that you use to remove the neurotransmitter? So this is the picture that we've been looking at, right? We had an action potential, which is number 1, right? Number 2 was the voltage-gated calcium channel, which moved calcium into the presynaptic cell. Number 3 is exocytosis of the neurotransmitter. Number 4 is causing the postsynaptic cell response, whatever it is, okay? But look what has to happen after that. So 4 and 5 is postsynaptic cell response. We have to get rid of the neurotransmitter so that the cell can respond to the next release of neurotransmitter. How do you do that? Some neurotransmitters just diffuse away. But that's not in synapses that really need to be used frequently because it's not a very effective removal mechanism, right? So some of them do just diffuse away. But there are a couple of other mechanisms. Other mechanisms are sometimes on the postsynaptic cell membrane, there is an enzyme that changes the shape of the neurotransmitter so that it can't cause a response anymore. And you already know one of those enzymes, right? Yeah, acetylcholine. So let's look at, acetylcholine esterase. So let's look at this since we already did it. This is a neuromuscular junction, but it's still a synapse, guys. So we caused the desired cell response, right?
Action potential is generated.
And then what we are going to do is we are going to break it down, right? So that the postsynaptic cell can feel it when we do summation. Otherwise, it would just have the same response from a single release of neurotransmitter. Does that make sense, guys? Okay, so sometimes it's an enzyme that causes a breakdown or a change in shape of the neurotransmitter so that it doesn't cause it anymore. And then the other way to do it, there's 2 other ways to do it. The other way is that sometimes the catcher rolls the ball back to the pitcher, rolls it back. Yes? So that you can reuse it. So you just reuptake it back into the presynaptic cell, again, to reinforce. Does it cause a presynaptic cell response? No. Okay. This is usually, by the way, guys, a pump that just pumps it back in. You repackage it so that you can use it again. Good? So that's a reuptake mechanism. And then the other way is not drawn in this picture because they didn't actually know about it when they wrote this book, which is freaking cool. So there's one other mechanism, and that is that sometimes astrocytes, remember our friend the astrocytes? Astrocytes play ball boy for all this. So I've got an astrocyte, which is just hanging out, and astrocytes like structural stability in the nervous system. It also assists in the blood-brain barrier. Apparently it has one other job. Sometimes the astrocyte can pull a neurotransmitter out of the synapse and hand it back to the presynaptic cell. We didn't even know that before, so we ain't got any drugs that work with astrocytes at all for that. Okay? Does that make sense? So basically, these are the mechanisms of removing the neurotransmitter. So let's talk about a couple of these that may end up being relevant to you. So neurotransmitter diffuses away. We don't have any drugs that treat that. It just does its thing. We do have drugs that treat the enzymes and treat the reuptake mechanism. So let's talk about that reuptake mechanism. And we're going to talk about this in more detail later. But of course, you guys know that serotonin is one of your mood-regulating neurotransmitters. It's primarily a CNS, yes, central nervous system, mood-regulating neurotransmitter. Low levels of serotonin can manifest as anxiety or depression. Usually, it's a gender predisposition. Females, low serotonin is more likely to manifest as anxiety, and males, it's more likely to manifest as depression, but there's overlap between the sexes and genders, or whatever you want to call them in that instance. Don't know which would be the appropriate term in that instance, sex or gender. One of the ways that we can treat anxiety and depression, give it a shot anyway, is to allow for whatever serotonin, let's pretend this is a serotonin synapse, is released to stay in the synapse longer. And doesn't that kind of pretend like it's frequency coding? Like it's more serotonin being released if it stays in the synapse longer? Yes, doesn't that? So what we do is we inhibit the reuptake of serotonin at those synapses with a category of drugs called SSRIs, selective serotonin reuptake inhibitors. And sometimes SSRIs can cause a relief from depression and anxiety. Now, ideally what you would do is say, hey, you're depressed, you should do all of the things that will make you make more serotonin. You should exercise more and get more sleep and stop hanging out with assholes and have more sex. And then you're like, I don't have time for any of that. I would like a pill, please. So does that make sense to you guys? Okay, go ahead.
So SSRIs inhibit the reuptake of serotonin.
Yeah, but we're going to talk about them in detail when we get to serotonin. So I was just giving you an example of those. And then the other drugs that we have that manipulate this specifically are we have drugs that block the breakdown. Yeah, block the breakdown at certain synapses. And we're going to run into those too. There are drugs that block the breakdown of, for instance, dopamine and norepinephrine and epinephrine. And so you can block the breakdown. And that would mimic the appearance of more dopamine, more norepinephrine, more epinephrine. Go ahead.
Would that be used for like, what's the disease when you have the dopamine slow and cause like, I'm sorry, it's kind of like MS, but it's.
Parkinson's.
Yeah.
Yeah, we don't have anything that can get to that region of the brain. In theory, it would work, but we don't have anything that can get to that region of the brain. Okay, so we're going to run into drugs that do several of these things as we go through. So as we go through a tour of the neurotransmitters, we're going to meet the neurotransmitters. And then if there is a disorder or a drug that you've already experienced or heard about, we're probably going to talk about it. Right? So that you can understand, what does alcohol have to do with GABA? What does cocaine have to do with dopamine? Yes? Does that make sense? So you're going to end up learning neurotransmitters and also learning applications of neurotransmitters. Does that make sense? Okay, so these are the ways of getting rid of the neurotransmitters. So now let's just deal with one last thing before we actually get to the idea of summation. So what I have been showing you is a one-to-one relationship of presynaptic to postsynaptic cell. Where are you? So one pre, one post. That is not how the CNS works. The CNS does not generally have one connection to a single neuron. In the central nervous system, one neuron, now this is going to look crazy, but it gets crazier when I tell you. One neuron has up to 15,000 synapses. 15,000. Okay, but they all work along the same story, right? So you can tell the one synaptic story, and then you can try to understand how that would be extrapolated to 15,000 synapses. So the thing that I need to tell you is that the one-to-one relationship really doesn't occur. What generally occurs is one or a combination of these two things. Often one postsynaptic cell will have multiple presynaptic cells converging on it. This is called convergence. Does that make sense to you? That concept should make sense to you. In this picture, some are red and some are green because the red ones are going to be the red ones are going to be inhibitory synapses and the green ones are going to be excitatory synapses. Okay? So what this guy does will be a combination of how much excitation and how much inhibition it's got. And I'm going to teach you that in just a second. But basically, you can have some cells trying to depolarize and some cells trying to hyperpolarize, and the cell's like, I don't know what to do. Generally, what it does is whichever one is more persistent, right? Because sometimes it's just going to go inhibit, screw it. And then the excitatory ones are going to keep going. I know you experience this when you're trying to get your butt out of bed in the morning and you've only had 3 hours of sleep, right? You've got some of them that are going, go back to sleep, you can't live like this, right? And the other ones are going, you already paid for the damn class, get up, right? And then what happens depends on usually which ones are most persistent. So this is called convergence. Lots of input lots of pre to one post. And then this is called divergence. One pre will diverge and stimulate a lot of post. Does that make sense? This is called divergence. Okay, now what is happening, for instance, in your cerebral cortex? Convergence or divergence? Yes. All of it is happening in your cerebral cortex. You have one that is getting a lot of input and one that has a lot of output. But an even better example is your hypothalamus. So if we talk about the hypothalamus, in addition to all of the receptors that it must have for hormones, right? Your hypothalamus has input from a whole bunch of sensory areas, metabolic areas, light, temperature, right? It has all of those inputs to decide what kind of output. You guys with me with the hypothalamus? Lots of input to the hypothalamus, right? Temperature, metabolism, light, all that kind of stuff. But then also the hypothalamus is also going to control a whole hell of a lot of things. It's like a little freaking Napoleon, isn't it? It's like the hypothalamus is going to branch out and control a bunch of other regions of your brain. So the hypothalamus would probably have both convergence and divergence. Does that make sense? Questions? You guys are being too quiet today. Frightens me when you're quiet. And Anna needs some coffee. I'm going to teach you how coffee works in just a second, too. Okay, so FYI, so what is going to happen is the axon hillock right here doesn't care how you got to minus 55. If you get there, you get an action potential. So what the axon hillock is going to do is it's going to sum all of the excitatory and inhibitory inputs, and if you get to minus 55, you get an action potential, and if you don't, you don't, right? It doesn't really matter where it was excited or where it was inhibited. None of that matters. FYI, interesting tidbit is that I told you that you could form new synapses, right? And this one could, for instance, branch out and form new synapses. It seems as if most of your synaptic formation and that synaptic plasticity happens when you're sleeping. So this one was super interesting for me to discover. So for years and years, we've known that your brain has almost the same metabolic needs when you're awake versus asleep. Like it uses almost the same amount of oxygen and glucose when you're awake versus asleep. And people are like, what the hell are you doing in there, brain? And it wouldn't tell you. So it's interesting because when you're awake, it's doing a lot of conscious sensory processing, right? And a lot of voluntary motor activity. Yes, like your cerebral cortex is doing that. But when you're asleep, it's doing very little of either one of those. And yet it's still gobbling up oxygen and glucose. So what the hell is it doing? Well, it seems as if primarily what it is doing is taking the dump truck of information and experiences that you had when you were awake and forming new synapses based on those experiences. So one of the things that we've learned about that is that sleep disturbances tend to manifest with memory disturbances. Alzheimer's, long before it manifests as a memory problem, it manifests as a sleep problem. And we'll talk about Alzheimer's several times as we go through here. Is that okay? Okay, so now we are going to talk about summation, and then we'll get into the neurotransmitters. Okay, so how do we get this postsynaptic cell to an action potential? Generally, here's what doesn't occur. One action potential here does not cause an action potential here. Generally speaking, you are going to have more than one action potential in the presynaptic cell to get the postsynaptic cell to threshold. And so that means that there is a delay right here in the synapse, right? There's going to be a delay. This portion right here is, as I mentioned, the slowest portion of the nervous system. It is called synaptic delay because often this one has to release neurotransmitters 1, 2, 3 times before the postsynaptic will get to threshold. This synaptic delay is usually around 0.2 milliseconds to 5 milliseconds, but there is no way in hell you could ever notice it because your noticers work with that synaptic delay. So there's like no way you could ever notice 0.2 to 5 milliseconds. So this is going to require what we call summation. So I want to talk about 2 different ways to get that postsynaptic cell to an action potential. You ready? Okay. So I want you to orient yourself to this figure before we start talking about it. So what we have is 3 presynaptic cells, 1 post, yes? And they are color-coded. These two are going to cause EPSPs, yeah? And this one's going to cause an IPSP, right? But in this first example, we're only going to use A. We're not going to use B and C, right? Even though B and C exist, we're not going to be using them at this point. So my question to you is, how can I get that postsynaptic cell to an action potential just using one presynaptic cell? What can you do? Frequency. It's frequency coding. It's always frequency coding, right? So if I stimulate A and then let me completely repolarize, then I don't get any summation, right? But what if I stimulate A very frequently, right? Then what's going to be happening is I'm going to add to the graded potential. Yes, I'm going to depolarize. I'm going to depolarize again until I get to an action potential. Go ahead.
This is able to happen because it's not an action potential, it's graded potential, right?
Great question. I'm glad you asked that. So if you had an action potential here, then you would be in absolute refractory period. But refractory periods do not apply to graded potentials. They only apply to action potentials, right? So I'm not in absolute refractory period until I've opened my voltage-gated channels. Yes? Okay. So I can do, this is called summation or temporal summation. Why temporal? Because it's time-related. Temporal summation. And it's really similar to summation in muscles, right? Because in muscles, what you do is you stimulate the muscle. Only in the muscle, you're getting an action potential every time you're just summing calcium release to get a stronger contraction. Here, you're not getting an action potential. You're summing graded potentials to get to threshold. Does that make sense? But the presynaptic cell is doing the same thing, isn't it? Right? The presynaptic cell is behaving the same with muscle summation and temporal summation in neurons. Yeah, it's doing the same thing. Everybody good with that? Okay, go ahead.
So should I cross out many action potentials are generally required for presynaptic neuron to release a neurotransmitter and do graded potentials, or is that?
Oh, did I? No, you have to, in the pre, it has to have many action potentials.
Oh, so it's still not many action potentials, but right
In the
now
pre.
we're talking about
Yeah, so many, where does it say that?
the beginning.
Okay. Many action potentials in the pre
Okay.
are required to release enough neurotransmitter to reach threshold in the post.
Okay.
Yes? So the pre is having an action potential every single time because it's releasing neurotransmitters, right? So here, action potential, action potential, action potential causing this, yes? The post, I haven't gotten to threshold yet.
Okay. The post is integrated.
Yes, the post is doing the summation. Got it? Okay, good question. Okay, so there is another way that you can do summation or get the postsynaptic cell. So in this one, I was only using one of the 3 synapses, right? But I could also use more than one at a time. This is a different type of summation. It's called spatial summation, because I'm going to use more than one space or more than one synapse. So look, watch the diagram. So again, A is excitatory, B is excitatory, C is inhibitory. If I use A by itself, no threshold. B by itself, no threshold. C by itself, definitely no threshold, right? But what if I use A and B simultaneously? Yeah? A and B simultaneously is spatial summation. I'm depolarizing here, hopefully collectively. That will cause enough of a depolarization that the axon hillock will depolarize. Got it? Okay, so this is spatial summation. Now, can you sum to 0? Well, yeah, we've taken math, right? If you excite and inhibit simultaneously, the cell's going to go, I don't know what the hell you want me to do. You better work it out yourself, right? Okay, so this is spatial summation. So logic question for you. Okay, in this instance, right here, this postsynaptic cell, what is the fastest way you can think of to get that postsynaptic cell to an action potential? Spatial. Yes, exactly. Use A and B simultaneously and really frequently. Yes? Does that make sense? Okay, and tell C to bug off. Right? Does that make sense? So you don't have to use them exclusively. Those are just 2 ways that you can get to an action potential. Do you understand the difference between the terminology of temporal summation, as in time? Yes, temporal means time, implying frequency. And spatial summation implying more than one space or more than one synapse. Does that make sense?
Yeah.
Okay, all right. Questions? No? Okay. All right, so let's talk about what happens if your synapses are not doing what they're supposed to do, and then we'll start to get into specific neurotransmitters. So it happens to every damn one of us, at least at some point in our life. Our synapses are not doing what they want, what we want them to. It may manifest in the central nervous system as anxiety or depression or OCD or ADD or just complete lack of joy in life. So how the hell do you get these things to behave? Well, there are some theoretical ways, and then there are some actual ways that we can intervene. And as we go through each of the neurotransmitters, I will tell you the drugs that exist, right? Ideally, of course, if this was not releasing enough, what we could do is say, hey, make some more or release some more. And we don't have any drugs that do that. We don't have drugs that can directly cause you to, for instance, make more serotonin or release more serotonin. Okay? Even if we did, serotonin is used about 6 different places in the brain. It not only is a mood regulator, it also makes you hallucinate and works on your brainstem and your sensations. So you'd have to be super selective about making you make and release more serotonin in one little place. So this would be ideal. We don't have a whole hell of a lot of control over this. This is some of the things that we can try to get you to do by changing your life, right? Like have more exercise, have more sex, don't hang out with jerks, right? But we don't really have any drug interventions that can do that. We do have drug interventions that can do this. Interfere with removal of the neurotransmitter once it goes into the synapse. And we will talk about these as we go through. SSRIs are selective serotonin reuptake inhibitors. You would use those if you thought you needed more serotonin. SNRIs are selective norepinephrine reuptake inhibitors. You would use those if you thought you needed more norepinephrine. MDMA is ecstasy. It does a couple of things. It causes a massive release of serotonin and inhibits reuptake of serotonin. And it's actually being used again as a therapeutic drug, and we'll talk about how that works. And then MAOIs are monoamine oxidase inhibitors, and they actually inhibit the removal of dopamine, norepinephrine, and epinephrine from synapses. So these categories will all go through as we go through the specific neurotransmitters. And then there are also other things that exist. If I, for instance, had too much of a neurotransmitter and didn't know how to get rid of it, I could use a blocker, yes, that would block its impact on the receptor. Those are called antagonist drugs. Does that make sense to you? Okay. So you could use an antagonist. An antagonist binds to the receptor but doesn't cause a postsynaptic cell response. So it's basically a blocker. Does that make sense? Okay. An agonist binds to the receptor and causes a response that is similar to the neurotransmitter. Sometimes it's not exact, but it's similar. Okay, so agonist, antagonist. Let's look at a couple of agonists and antagonists real quick. Okay, so an agonist. Tell me, guys, just to make sure that you're understanding what we're talking about. You might use an agonist if you had too much or too little of the neurotransmitter. Too little of the neurotransmitter, right? You'd put something in there that mimicked the effect of it. Okay, so regardless of the treatment drugs, let's tell you a drug that people do take in that is an agonist. So nicotine. One of the reasons that people like nicotine, and I am not the kind of part of the reason that I don't teach high school, for instance, is I'm not going to just teach you drugs are bad without telling you why the hell people want to do them in the first place. Because I'm not the kind of child that would have appealed to. You say it's bad, I say I am now very curious. Okay, so why do people like nicotine? Well, initially what happens with nicotine is that nicotine is an acetylcholine agonist. Acetylcholine increases short-term memory formation and mental acuity. So when you take in nicotine initially, it makes you feel like you're firing on all cylinders. You will very quickly downregulate that response and tap into the addiction pathway, and so it won't do that for you for very long, but initially it does that for you, right? So nicotine is an acetylcholine agonist. It behaves kind of like acetylcholine in the central nervous system. Does that make sense to you? Okay. And I always wondered why the hell people did it. Both of my parents died of smoking-related disorders. And I don't want Java. And I stay the hell away from it because I know that it's not good for me specifically. Antagonist drugs or chemicals occupy the receptor and block the response. Okay? Would you theoretically use these when you had too much or too little of the neurotransmitter? Too much, okay. Does it surprise you that a lot of poisons are antagonists in the nervous system? Like a lot of neurotoxins that can kill you are antagonists. So let's talk about a couple of them. You ready? So let's talk about fugu. Tetrodotoxin is from fugu, which is the pufferfish. Okay, so pufferfish pretty much are trying to do everything they can to tell you that you probably shouldn't eat them. And yet, humans are like, you know that pokey thing that has poison? Pretty sure I'm going to eat that. So it's a defense mechanism in the pufferfish, right? So they have this gland that produces a neurotoxin. And this neurotoxin, what it actually does is it blocks acetylcholine from binding to skeletal muscle fibers. And so you can have respiratory arrest with the diaphragm while you're completely conscious because it doesn't do anything to your cerebral cortex. So you're like, can't breathe, I'm definitely dying, definitely dying. So, but of course, people eat it because we are just freaking bored as hell after the age of like 17. We're like, seriously, is this all my brain does? And so we decide to eat poison things. And apparently it's also delicious, but I've never had it. I tried to get my husband to let me have it in Japan when we were there. He's like, you didn't bring $500 extra, shut the hell up. So it's really expensive. You can't get it in most places. You can get it in Japan, a couple places in California. And the chef has to be trained really, really well to make sure that you remove the gland. Now, why do people like it? Well, apparently. Has anybody tried it?
No, I don't think
You don't like
No.
What? Apparently, it not only is delicious sushi, but also gives you a little tingle when you're eating it. And that tingle is, you almost died. But you weren't bored, were you? No, you were not bored at that particular moment. And hell yes, I'm going to try it at some point. Okay, so that's tetrodotoxin. Saxitoxin is in red tide. Have you guys heard about the red tide that's going on the Gulf Coast of Florida right now? So red tide is this protozoan that increases in population during certain environmental conditions, and it's going to become far more common, so with global warming. And when it has a bloom, what it does is this protozoan produces a neurotoxin that gets into every single thing that eats it, kills off fish. You can't eat a damn thing out of the ocean. You can't even go into the ocean. If it gets concentrated enough, it starts killing off marine mammals as well. So almost the entire Gulf Coast of Florida has had swimming bans and fish bans for most of the last 2 or 3 months. So that's red tide. That's what it's called. Just go look it up today. It's kind of interesting. Honeybee venom it's an antagonist, but it just stays local, so it doesn't like do much, right? It's just like a little local antagonist. Black mamba venom is an antagonist again for acetylcholine, so it'll kill you by respiratory arrest. Just a second. And then sea anemone is also an antagonist for a neurotransmitter, but it stays local too. So it doesn't usually kill you because it's just like locally doing that. Go ahead.
This might be a stupid question. Is black mamba like a fish?
No, black mamba is a snake.
Oh, a snake.
Yeah, snake. Black mamba.
Or did I say fish? I meant spider.
No, black widow is a spider, and we're going to talk about that too. But black mamba is a snake. It's also the name of my husband's black fortune. And my car is Judy. Julie. Like, why is my car not as cool as Black Mama? I don't know. Because my children named it, and they think he's cooler than I am. Bastards. He is. Okay, and then let's talk about caffeine, our friend. Caffeine. I love you, caffeine. Caffeine is an antagonist, but I want you to listen to how caffeine works. Caffeine is an antagonist to an inhibitory neurotransmitter. So what does it do? Yeah, it blocks inhibition in the central nervous system. So what do you get? Excitation in the central nervous system. That's how caffeine works. It's an antagonist to an inhibitory neurotransmitter called purine. It has other PNS effects, but that's how it actually wakes up your brain. That's what it does. And so when you're trying to go to sleep and you decided to have a Red Bull before you go to sleep, good luck with that. It's not going to work at all. Okay, so now we are going to start our little tour of neurotransmitters. Do you guys have any questions before we start our tour? No? Go ahead.
What's the neurotransmitter that caffeine blocks?
Purine. Yeah, we're going to cover it again as we actually get into purine. Okay, so here we go. So what we're going to do is a tour of neurotransmitters. You are not going to learn all 60 of them. And new neurotransmitters are being discovered. You are going to learn the neurotransmitters that you will probably mess with to try to stay sane and functional, or that you will run into clinically. So those are the ones we are going to talk about, or that I know that you're going to run into throughout the rest of your training for whatever you're going to do. We won't do all of them, so please be open to new information and don't be one of those people who finishes their education and never reads a damn thing, because you're going to be a terrible medical professional if you do that. Okay, so you guys tell me, before we get into the specific neurotransmitters. Neurotransmitters have to meet 2 criteria. They're chemical messengers released from where? Cell. Chemical messengers released from a neuron close to what? Close to the receptors. Yeah, that's the criteria you need to meet in order to be a neurotransmitter. And these are basically the language of the nervous system. This is how the nervous system communicates. Now, we will have neurotransmitters that are CNS, that communicate between neurons within the brain and the spinal cord, and that are PNS, that communicate between neurons and other things in the peripheral nervous system, usually muscles or glands. And some neurotransmitters will have CNS and PNS function. Does that make sense to you? Okay, so if I say it's a CNS neurotransmitter, right, then that is functioning in the brain somewhere, or the brain and the spinal cord. If it's a PNS neurotransmitter, it's functioning between a neuron and like skeletal muscle or something. Okay, which one do you think we know more about, CNS or PNS neurotransmitters? PNS, yeah. We know a hell of a lot more about the PNS neurotransmitters, even though they're not the most interesting ones, because you can totally jack with them without killing somebody, right? Like you can manipulate it. When you did the muscle lab, you were specifically causing PNS neurotransmitters to be released, but I'm not letting anybody mess with this but me, right? And you can't do a whole hell of a lot of experimentation. So we do know some about CNS neurotransmitters, but probably not the answers to all of your questions. And they often, of course, bind to receptors that are ion channels and start an EPSP or an IPSP. There's about 60. Characteristics of a neurotransmitter. Most are relatively small, like low molecular weight. And that means that they are going to diffuse a little faster. Like here's acetylcholine. Notice that it is not a string of 150 amino acids, right? It's relatively small. It's going to diffuse relatively quickly across the synapse. And they're basically stored for release. And they are going to be fast acting, right? And usually, even though you have one neurotransmitter, it might have more than one receptor subtype. So in order to figure out what the neurotransmitter is going to do, you have to figure out what tissue you're talking about. So acetylcholine could be excitatory on this tissue because it binds to that receptor, and inhibitory on that tissue, right? Does that make sense? Okay, so what we are going to do is we're going to try to get a decent understanding of the most important, well-known neurotransmitters by doing this tour of neurotransmitters. And we're going to start with the one that we know the best and have known about the longest. And you do have to categorize these. We're going to categorize them based on their chemistry, not because I know you guys love chemistry and love when I talk about chemistry, but because if neurotransmitters are really chemically similar, like dopamine, norepinephrine, and epinephrine, then a lot of times they use the same reuptake mechanism or the same breakdown mechanism, and you can use drugs that treat all of them at the same time. So that ends up being clinically significant, right? So if they're really similar to one another. So we're just going to start with acetylcholine, because that's the one that you are already familiar with, because we use acetylcholine to stimulate skeletal muscles. It is going to go into a category... It's going to go into a category of neurotransmitters called choline derivatives. And it's the only one that's in this category. I mean, it's not the only one that exists, but it's the only one you're going to learn. And acetylcholine is derived from a water-soluble B vitamin called choline. You just like slap an acetic acid on it, and then you got acetylcholine. Okay? So... the only one you're going to learn is acetylcholine. And again, I never will ever make you even recognize, much less draw these. That's what acetylcholine looks like. Okay, so I want to talk briefly about acetylcholine. It is a usually excitatory neurotransmitter. I only say usually because most receptors for it are excitatory receptors, but not all of them. Okay, so it's usually excitatory. And it has PNS functions and CNS functions, Okay, so it's acetylcholine. It is PNS and CNS. Okay. PNS functions, you already know a lot of them. You know that it is the neurotransmitter that is always used for skeletal muscle, yes? Okay, so the entire somatic nervous system uses acetylcholine, yes? And half of the autonomic nervous system also uses acetylcholine. So really, for those of you who care, and we'll talk about this in the next 7 minutes, it's the parasympathetic branch of the autonomic nervous system that uses acetylcholine as its final neurotransmitter, but you don't have to know that yet. So basically all of the somatic nervous system and half of the autonomic nervous system. And then CNS functions, I already told you this, but the CNS function of acetylcholine, simplified, is that acetylcholine helps with short-term memory formation. So when you experience something and stick it into your short-term memory, that is primarily acetylcholine working in there. It's not long-term memory, but you have to get things into short-term before they can get transferred to long-term. Okay, now let's just do a little terminology. Okay, so acetylcholine, it's PNS and CNS. I'm trying to show you what you will need to pull out about every neurotransmitter. Terminology. The fast way to refer to anything related to acetylcholine is to call it cholinergic. So look at this picture. The presynaptic cell releases, the postsynaptic cell responds to, the receptors bind to, the reuptake mechanism or breakdown mechanism are all related to acetylcholine. So the terminology, to say that really quickly, is the whole blasted thing is called cholinergic. Okay? So presynaptic cell, cholinergic. Postsynaptic cell, cholinergic. Receptor, cholinergic. Reuptake mechanism, cholinergic. It's just a fast way to say releases, responds to, binds to, whatever the hell. Cholinergic. Okay? And then let's talk about a couple of receptors. The receptors that we are going to run into for acetylcholine are nicotinic, specifically nicotinic cholinergic, and muscarinic. Let me tell you how those things are named. So, a lot of times we knew about drugs before we knew about neurotransmitters or receptors, because we have been doing drugs for a long time, long before neuroscience was a thing, when they thought it was just all magic. Okay? The receptors that we found that bind to nicotine and also acetylcholine, what did we name them? Nicotinic, yeah? Nicotinic, does that make sense? So a lot of times the names for the receptors have something to do with history, and a lot of times have to do with like somebody dying of something, or somebody snorting something, or somebody smoking something at some point in the past. So nicotinic cholinergic receptors bind to nicotine and acetylcholine. That's how we found them. And eventually we were able to actually dye nicotine and watch it go to the receptors, right? Like make it fluoresce and watch it go to the receptors. And then muscarinic cholinergic receptors bind to acetylcholine and a mushroom poison that kept killing people, and we couldn't figure out why for a while, called muscarine. Okay? And we're like, oh, that's the one that kills you if you eat that mushroom. Okay? So nicotinic and muscarinic, and we'll run into those receptors as we go through the rest of the semester. And then the last thing that you need to know about neurotransmitters, when appropriate, it's not always appropriate, is drugs and diseases related. And we already know these. We don't know all of them, but we do know a drug that is an acetylcholine agonist. What is it? Nicotine, yeah? And we know a disease that causes a loss, well, maybe you don't know this one yet. A disease that causes a loss of acetylcholine so you can't form short-term memories. And it's Alzheimer's, yeah. Alzheimer's, yeah. So with Alzheimer's, at least in the early stages of Alzheimer's, not in the late stages of Alzheimer's, somebody might be able to remember their wedding 60 years ago, but they can't remember if they took their meds today, which is a big deal, right? Because you can't be compliant with your meds if you don't remember if you took your meds. Does that make sense? So that ends up being a big deal. So that's Alzheimer's. And then there's one last little drug. You may or may not care about this one. I don't even usually ask about it, but I like it because it's like a mystery story. Curare? Have you guys ever heard of Curare? Yeah?
What's in the last minute?
What?
It was briefly in the last minute.
Oh, yeah. Curare. Yeah, I didn't talk about that. Curare is a drug that is derived from a plant that I think is in South and Central America, but I'm literally always lying when I talk about geography because I don't know where anything is in the world except Texas. Would you like to know where Texas is? Because that's where it's like everything else is like other places. No, I don't think that way. That's just the way I was educated. So curare is from South and Central America. It's a plant derived. They use it on blow darts and arrows. And what it is it's an acetylcholine antagonist that will block acetylcholine from binding to the receptors. And you can use it to kill other people or other mammals if you want to. I learned about it when I was a kid because I read a whole bunch of mystery novels as a kid because I was a super duper dork and still didn't like people very much. So I read all of Agatha Christie by the time I was like 8. And we have some really fantastic ways to kill each other. And curare is one of them. So here's what curare does. If I shoot Mike, it binds to his acetylcholine receptors on his diaphragm and makes him basically die because of respiratory arrest. Yes? Or if I want to eat a monkey and the monkey's in the tree and I want to climb the tree, thunk, just wait a second, the monkey will fall out of the tree because his muscles will be temporarily paralyzed in the diaphragm. Apparently it has a relatively short half-life, so you could still eat the thing. Or you could have some migratory mammal that you didn't want to chase for very long. You could just thunk it and then wait for it to fall over. Okay, so that's what curare is. Okay, so next, what we will do, and I'm not going to start on it right now, is we're going to start the other categories. Biogenic amines are going to be next, in which we get to talk about your happy chemicals, dopamine and norepinephrine, and we'll talk about serotonin too. Go ahead.
I don't understand the subtypes and why they're important.
Oh, well, they won't be important yet, but once we get into cardiac muscle, I'll be able to tell you that this is an ectocentric nerve receptor, and that smooth muscle has a muscarinic, and that's why they behave like the same neurotransmitter is different on the two.
Oh, how they react.
Yes, exactly. So it's going to end up fleshing out as the rest of the semester goes on.
So should I pay less attention to why these are things and just understand their
Yes. Just understand the categories exist right now, and then later on we'll flesh out what that existence means. Yeah. Hi.
I got a person to tell me what testosterone cycle.
Oh, yeah.
I don't know what any of those mean, but maybe you know some of them.
Testosterone proper. I wonder what halotestin is and winstrol. What the hell? Don't tell me who it came from.
I
Hi.
have to fill out this bottom row. It's just for the Creo program.
Yeah, let me record. Let me stop
Do
the
you
recording.
have, airdrop or anything?
Yeah, I do. Uh-huh.